joining us today to learn about early detection invasive species. All right, so I am Brittany Hernon, the West New York PRISM Early Detection Project Manager. So West New York PRISM is a partnership for regional invasive species management. And just in case anyone is not familiar with the PRISM network, there are 8 PRISMs across New York State, and West New York PRISM covers the 8 westernmost counties. So we work with partners to carry out invasive species management with the goal of protecting local terrestrial and aquatic resources. And I also put our website up there, which is westnewyorkprism.org. So if anyone wants more information about what we talked about today or other invasive species, you can check out our website. Just a quick review. Invasive species are not native to an ecosystem, and they also cause harm to the economy, environment, or human health. And usually it's multiple aspects in all 3 of those. For example, aquatic species can cover a lake and decrease recreation and tourism and have economic impacts. Other species can outcompete with native ones and alter biodiversity and have environmental impacts. And even in the case like giant hogweed, they can injure people upon contact and cause problems for human health. Today, we will focus on early detection invasive species. So these are present in the region, but they're not common or widespread. And so they're only found in a few locations, and they have the management goal of eradication. If you come across any of the species we discussed today, ask that you follow our easy reporting protocol. So if you like to go out hiking and look for species while you're on the trails or things like that, it's very helpful. You can find our early detection reporting protocol on the website, westnewyorkprism.org. So it's under the invasive species tab in the early detection section. And so it's easy to find and easy to use. It's very user-friendly. And so if you find one of these species and you think it's an early detection species, just take down a bit of information. So the size, you can take down acreage, the length that it stretches along a trail, something so that we have an idea to get a picture of how the species looks, and then also location. A GPS coordinate is great, an address, things like that, so we could go find it again. And then just some clear photos, really, close-ups of the plant, different features that helped you identify it, whether it was the leaves, the flowers, things like that. And then take all of this information and enter it into IMAP Invasive. So this is an online database where you can report invasive species. You can see where other invasive species are present, very easy to use. There's even an app that you can put on your phone. So while you're out on the trails, you could just take this information and send it right to IMAP. Or if you're unable to use that, you can send all the information to West New York PRISM at buffalostate.edu, and we will get it. And so when this happens, when things are reported to IMAP, I'll get an alert in my email, or we check the other email, and West New York PRISM will use the photos to confirm the species, or go take a trip out in the field and confirm the species on the ground and begin working on a management plan for this early detection species. So the first species we'll talk about is Slender Falls Brome. This bunch grass is native to Eurasia and North Africa. And as you can see from the picture, it forms these very dense clumps and creates monocultures. And if you look, no other species are really growing in that understory there except this grass. Each of those clumps in the picture produces hundreds of seeds each year, which get carried to new places on clothing, on animal fur, in mud that's caked in tires, things like that. And the species does well in many habitats. It can do, pretty dry areas, pretty wet areas, full shade, full sun. And it is not really eaten by wildlife or livestock. So essentially when the seeds make it to a new area and grow, the plants just kind of go unchecked. And this species is a perennial grass, and it produces flowers towards the end of June and mature seed in July. So if you are out on the trail and would like to try and identify the species, it is a bright green bunch grass. So this sort of clumping behavior is a good way to identify this grass. And then also if you look, the stems and nodes are very hairy. Put a little identifier on the screen so you can see where the node is, but hairy stems, hairy nodes, and then the leaves are flat and they arch out from the center of the plant and are also hairy. So really, these bunches of grass that are hairy is a good way to tell, and the bright green color. The plant also has flowers that are drooping and again arch out from the plant. And the seeds have these long awns, which again, I marked that on the screen. And so this is the part which helps them attach to your clothing or pet and wildlife fur and just move to a new area. So WNY PRISM and the Great Lakes Slender Falls Brome Working Group wanted to test out some different methods to determine the best way to get rid of this grass. As I mentioned, it's an early detection species. It's only found in a few areas, and so we wanted to see what works so we can try to move towards eradication in the West New York PRISM region. And so we used some infestations at Genesee County Park and Forest and Letchworth State Park to establish plots, collect data, and then implement treatment. And we also worked alongside some SUNY Brockport researchers at Burgeon Swamp to establish some plots there and collect more data during their management project. And essentially we went to infestations, we divided them up into different treatment sections, established plots within those sections, and then looked at changes in Slender Falls Brome percent cover and density over time, and also kept track of species in the plots. And so, as you can see right here, our first method was manual removal, and we went out with shovels to dig up the big plants and make sure to get the plant and the root and contain it in landfill-bound trash bags. And so essentially the first year we were out there with shovels working with these really large clumps. Then in the second year, as you can see in this other picture with all these people kind of on the ground, we basically crawled around looking for seedlings that grew in. So then we had some data to look at and assess this method, and it did significantly reduce percent cover, but it is very tedious and also detrimental to soil. So it is really recommended for infestations that are, less than half an acre. If it's greater than that, you might want to look into a different management method. And just some information about this treatment method. We worked in Letchworth State Park, and so in that area, in the first year, we collected 25.5 bags, and it took us 39 hours. And then we went back the second year, and we only collected 7 bags, so we dropped down from 25 to 7, which is good. That's showing the first year we had these really big plants. The second year we had these tiny little seedlings. So we removed the big plants, and so only seedlings could grow back in. But in the second year, it took us 47.5 hours to collect those bags. 39 hours the first year, 47.5 the second year, and we saw a similar thing at Genesee County Park and Forest. The first year we collected 7 bags of plants, took us 19 hours, and then the second year we filled 2 bags, and that took us 35 hours. I know it's not that exciting to, go home and say, oh, I spent this week pulling plants and we filled 2 whole bags, but it's important to take the time and get all of these seedlings and be really accurate. The more seedlings that slip through and become mature plants, they continue adding seed and just increase overall management time. So just something to think about when planning for management. The first year will not always take the most time. You might think that because there's more plants or bigger plants, but it's just important to be persistent, put the time and effort in to really see these positive results. And so I have some pictures of our manual removal. So you can see the first year when there were lots of plants, we dug up 1 large plant from this quadrant. And then the next year we went out and there were 23 seedlings in that same area. So this is good because we want to exhaust the seed bank. This species seems to only have a longevity of 2 to 3 years in the soil. But as I said, it takes a lot of time to find these little seedlings, pull them out. And so in some cases, people actually add nutrients and make sure the growing conditions are ideal when carrying out invasive species management. And so sometimes people make the growing conditions ideal so they can ensure seeds germinate and produce seedlings that can be removed. We can't see the seed in the soil, but we certainly can pull out tiny new plants before they become a big problem. So then the next thing we looked at is mechanical removal. So in this case, you take a weed whacker, a string trimmer, and you cut all of the plants as low to the ground as possible before they form flowers and seeds. So this was done in late June and late July. And this method is good to use while planning other management options. And at least in the thick monocultures that we worked in, it does not seem like cutting alone will eradicate the species. So it's a good way if you go out there and you find the species and you don't have time to treat it this year, you need some time to create a plan, cutting it will stop it from producing seed while you can determine what other option is best. And so as you can see in this picture, so I have a picture with the sign on the one side that shows the infestation when we started, and then the other picture with the quadrant is when we went out the second year. We still saw a lot of new growth and large plants when we went back out there. So like I said, this method is good kind of to prevent the species from adding seed and just buy some time to use another treatment option. We also used herbicide, so we applied glyphosate directly to Slender Falls Brome plants using a backpack sprayer. And so this method was incredibly effective. And so you can see in the one photo there are those big brown patches. So that is the dead Slender Falls Brome plant. And then you can also see some green in the area. What this is really showing is that, you know, the herbicide that we use is really targeted at the Slender Falls brome. And so it doesn't kill everything. It's a way to, you know, spray it directly on this plant and allow other plants to continue growing, and then hopefully have some native plants come into the area as the Slender Falls brome is filled over time. And so this shows how effective the treatment was after 1 year. So in the picture on the left, we have the one side where you can see some green grass on the ground. That's the side that was the cutting only. And then on the other side, it was treated with herbicide. And then in the quadrat, you can see again, this is the second year we went out. And so we have some seedlings growing in. When you use this method, you will have some dead plant material in the area, but herbicide treatment is nice because it's carried out in June. And so for people who have maybe a seasonal crew that's only there for a while, they can just go out and target the species in June and really get it under control. And then we tried a combination of cutting and herbicide treatment. So in this case, we cut in June and July and then treated in August. This method was also very effective. And so this kind of gives you the option to adjust treatment times. So with herbicide only, it must be done in June before the plants produce seed. In this case, the cutting prevents seed production, and so it can allow for later herbicide treatment. Maybe you need to get permits in order and need to do it later in the season, or that's just when you have the time or people available. So this is kind of a nice way to manipulate treatment so you can still get a handle on the species. And also there's less dead plant material in the area. So it is just as effective as the herbicide treatment alone. But in this case, you do need to spend time cutting the plants a couple of times in the summer and applying herbicide, so that's not always feasible. And so really what we saw is just, you know, to take into account the size of the population you have, the resources you have, and to look at some of these different management practices. So again, in the picture, we see lots of big plants the first year, and then only small Slender Falls brome seedlings growing back along with some other species the second year. This year, we will go out and focus on the infestations at Letchworth State Park and Genesee County Park and Forest to really tackle those areas, get them under control, and now we're moving from doing the research to actually implementing best management practices. And so in this case, we had a control area that we used to compare data. We'll be treating that, and we will also visit some of the smaller infestations at some parks nearby and manually remove those plants. So we have gone to some of these parks a couple years in a row and be able to dig up the Slender Falls brome plants, and we'll continue to monitor and just remove those seedlings, like I said, to really deplete the seed bank and move towards the goal of eradication. So a nice part of this research really was to see results so quickly. You know, for some management, it's very effective, but it may not be as dramatic or obvious over the first couple of years. So this is nice. It does make for some nice pictures to use in presentations like this. And so we're just excited that we can get back out there, continue monitoring the area, continue collecting data, and to continue treating these species. Also, we saw native species such as jack-in-the-pulpit, trillium, and milkweed, to name a few, grow back in some of the treated areas after the first year, which again is always nice to see. We'll continue survey work this summer and use a habitat suitability model to determine locations. And just a little side note on that, we surveyed an area called the Carlton Hill Multiple Use Area in 2017 and did not find Slender Falls brome, but this spot is very close to Genesee County Park and Forest. So in 2019, it came up as a sort of hotspot, a highly suitable area for Slender Falls brome on the habitat suitability model. And we did go back to the area in 2019 and find a few plants. So it's pretty cool to have this technology to guide our survey efforts and just really help us detect populations early. Just because you survey an area 1 year doesn't mean that the plants haven't spread there over the next couple of years. This one was really nice because we only found a few plants, so we could truly implement a rapid response effort by just digging up those plants immediately after we collected data. The next species is Japanese stiltgrass. As the name would imply, the species is native to Asia. And as you can see right here in this picture, it shows that the species forms these thick, dense mats that displace native species. It has also been seen that Japanese stiltgrass can inhibit germination of other species. So again, this changes the biodiversity, has a negative impact on the environment in the area. And also, so if you look at these pictures, there's a ton of these plants in that area. And so all of those plants eventually die back in late fall, and they form a very thick layer of thatch. The thatch takes a while to decompose and can also be a fire hazard. So this grass is pretty easy to identify in the summer around July and August. We have found it along Cazenovia Creek in East Aurora and West Seneca. Those are kind of the two big areas, but also some other parks like Chestnut Ridge and Franklin Gulf. It's not always along the creek, but it has been found there. It's also found along trails, and it also likes shade. If you enjoy hiking in those areas, definitely keep an eye out for this species. I have the picture right there, but you can see the leaves are 1 to 3 inches long. They have a pale green color. They're narrow. They have a lance shape and a distinctive silver midrib, as you can see in this picture. So this characteristic makes it very easy to identify and distinguish from other grasses. It does get tiny flowers sort of later in the summer, and also it has shallow roots, which is the picture on the bottom. This is another easy way to identify the species. So if you see a grass with a silver stripe that you can pull out of the ground very easily, it is Japanese stiltgrass, and you should report it to IMAP or let us know. That would be very helpful. So what can you do to manage this species? So as I mentioned, it's very easy to pull out of the ground. So for small infestation, hand pulling works very well. You can see here we have some happy smiling faces, hand pulling the species. It's not too bad to sit there and pull out this plant. It's a little bit better than some of the other ones where you have to go out with shovels or brush cutters. You can sit and just pull it out of the ground. So that works for small populations. And then in the case of larger infestations, herbicide treatment with glyphosate or some grass-specific herbicides is effective. And again, mowing can reduce seed production, but it has not been shown to be an effective way to remove populations of this species. And West New York PRISM has a couple of different projects focused on stiltgrass. So we will continue to survey along the creek to find new populations, and we will also create and use a habitat suitability model to determine and prioritize survey locations for this species this year. We need to know the presence and distribution in our region so we can effectively manage the species. So we will go back to the small infestations found over the last couple of years, continue to monitor the area, pull out the new plants. Last year, West New York PRISM went out and used herbicide at Hunter's Creek, so we are excited to get out there and assess how that worked this summer. So this picture that I have here is actually from Chestnut Ridge, where Japanese stiltgrass was found. It's just showing that the infestation continues past to the park boundary onto private property, which is a challenge for invasive species management. That requires education and outreach. In 2018, we sent out a postcard to areas near infestations in the town of Aurora and village of East Aurora. So you could see the card here. It describes the species, describes how to report it, and just encourages people to kind of go out, check their yards, and let us know if they found it. We may need to do something like this in the Chestnut Ridge area or other areas that we found where infestations continue onto private property. Something that we had in the works earlier this year was to train volunteers for a strike force, since this is a very easy grass to identify and also remove. It could be something fun for people to go out and do. At the moment, this has been put on hold because we are not encouraging large groups of people to gather together, but some things that we had talked about, we have a town supervisor from one area that's interested in coming out to see the plant, and we were also talking with one park. They said that they work with companies sometimes to have people come out as like a volunteer day as part of their work day to get people outside. So we do have some of these ideas kind of on the back burner, but we really hope to get that going in the near future. And so, again, it's a very easy species to identify. So if you are interested in being a part of this, you could definitely reach out to us, or it's just one to look out for while hiking and enjoying the outdoors. And again, if you have any interest in going out and looking for these species, please let us know, and we can provide you with more information on, what that entails. And so now we will turn the program over to our next two presenters to learn about Mile a Minute.
Hey, thank you, Brittany. Garrett, are you on as well?
I'm on.
Excellent. So thank you all for listening to us today. So Garrett and I are going to talk briefly about Mile a Minute in Western New York. Three things we're going to talk about. Briefly, we're going to talk about the characteristics and identification of Mile a Minute, the history of Mile a Minute in Western New York and when it was first identified in the region, and then our current efforts at research control and surveying. So Mile a Minute is in the buckwheat family. Its scientific name is Persicaria perfoliata. It's native to Eastern Asia and the Philippines. It was introduced in the late 1800s to early 1900s, and it was first found in Oregon in 1890, but that population did not establish. It was first established in 1946 in Pennsylvania from contaminated nursery stock. And from then, it spread rapidly and has created substantial infestations in New England and down towards south in Virginia, North Carolina, and Pennsylvania. So to identify mile a minute, you've got a few main characteristics to look for. So first of all, this is a climbing vine that is an annual vine. It's relatively weak. It's entirely herbaceous. It does not produce any woody material. So it will actually grow from the ground all the way up to 10, 15 feet in the air just within one growing season. It is this very, very bright light green color. It's typically lighter than most of the other plants in the forest and on the forest edge. It has triangular shaped leaves, which you can see in the leftmost figure. And the leaves are very weak and thin. So if you touch them, they don't feel very robust. On the stems, there are backward bending barbs. And these barbs actually act as little hooks to help it climb up on other vegetation and climb up on you if you happen to walk through it. One of the more distinguishing features about this vine is the distinctive ochrea at the nodes. So in the middle picture, you can see the circular leaf material that wraps around the nodes. That's called the ochrea. And that circular material at the nodes is at every node and goes all the way up the plant stem. The flowers are relatively inconspicuous. In fact, the flowers don't even open on most individuals. They stay shut, and the fertilization occurs via selfing. So there's no showy flowers to notice. And you can see what look like flower buds that will then transfer directly into ripe seeds and never actually open until the seeds are bright purple. So when the fruits are ripe, they're blue, as you can see in the middle picture. So on one inflorescence, you'll have fruits of different ages. So there's unripe fruit on that inflorescence. There's light pink colored fruit, which is getting towards ripe, and then dark blue fruit at the base. So the most common species that is mistaken for mile a minute in our area is bindweed. And bindweed has a similar growth form. It's also an herbaceous vine that will climb up and on other vegetation. Bindweed also is considered invasive and does have an ability to kind of overwhelm other vegetation and get tangled. But it has a few main differences that can let you tell it apart from mile a minute. So first is the flowers. So the flowers of bindweed are very conspicuous and large. So if you see these type of flowers, you're not dealing with mile a minute. Additionally, bindweed does not have the distinctive hooked barbs, so you would not be as likely to snag it on you if you're trying to remove it from an area. The leaf shape is a bit different. The leaves are sagittate rather than triangular, although the difference is pretty subtle to the untrained eye for people that are new to identifying plants. It does not have those round ochreas around the nodes, so the nodes are bare, and that can be a giveaway for the fact that it's not mile a minute. And then the fruits are not going to be those ripe blue fruits like mile a minute has. The fruits are capsules, and they do not look like berries. So the reason that this plant is so invasive is that it likes disturbed habitats and early successional habitats, which much of Western New York is currently in that successional stage. It thrives in areas with high sun and pretty good moisture levels, but it can go pretty far back into the forest, and you will find scattered individuals in clearings in woods. However, if you're going to survey for mile a minute, you're going to be wanting to be surveying on the edges of forest and in disturbed areas for those major populations. It grows extremely rapidly, which is why it's called mile a minute, up to 15 centimeters per day. My student, who's currently working on mile a minute in the greenhouse, was measuring plant height, and she got out early one morning, measured plant height, and by the time she left, after a 12-hour day in the greenhouse, she had to re-measure her plants because they'd already grown substantially. It is a prolific seed producer with extremely high seed viability, And one big concern is that even unripe seeds, so the fruits that are not ripe, will have seeds that still will successfully germinate. Germination is lower, but still has relatively high success, so you can't wait until the fruits are ripe to get rid of this problem species. The fruits are easily dispersed by birds, and the fruits and seeds can float for multiple days in aquatic areas, so this could also increase dispersal in riparian corridors. It's estimated that it's currently found in 20% of its potential habitat in the U.S. So this is definitely of concern. When it first appeared, scientists thought perhaps it wasn't going to be a big problem as far north as we are, but it's clear that mile a minute can do well here, and with changing climates, it's likely to become even more of an amenable habitat here in our area.
Okay, thank you, Katie. I'm going to take it from here and just discuss and replay the history of mile a minute and management here in Western New York. The first actual report that came in from this Western New York, Western Finger Lakes area was from a homeowner who was actually taking classes at Finger Lakes Community College at the time. I believe it was during one of Dr. Gilman's presentations. I'm not sure on that, but anyway, she saw the slide, learned what that weird plant was in her garden, and brought it forward to Dr. Gilman. They talked about it. He was pretty sure it was mile a minute. They decided to coordinate. Dr. Gilman coordinated with Hillary Mosier from Finger Lakes PRISM, and the two of them invited me to come along, and the three of us all together visited with the landowner, and Dr. Gilman was able to identify that it was mile a minute. So it was late September at the time when it was identified, and we got out there, and we all kind of looked at each other and go, all right, what do we do right now? Being that it was so late and being that the plant's an annual, we knew that controlling the actual plant itself didn't make much sense because it was going to die back and wouldn't be a problem until the following year. The problem at that time was the seeds. Because it propagates solely from seeds and it produces a tremendous number of seeds, we decided to focus solely on collecting and destroying those seeds. One or two folks working for Finger Lakes PRISM were out there religiously, and this is one photo in the bottom right of the corner of the screen here. This was from a gentleman named Dylan who bagged that many seeds in the course of a couple of weeks. And every one of those seeds is a potential plant, so that saved us a lot of potential headaches. It didn't get everything, but we were able to do some form of control that season. So over the course of the winter, between 2016 and 2017, we started making plans. I became the unofficial head of management here for the Geneseo infestation and began talking and collaborating with as many people as I could because I did not know what I was doing at the time. This was my first instance dealing with it. So we got talking, we got coordinated with groups. We had a lot of partners that were huge help between the Finger Lakes Institute, Finger Lakes PRISM, multiple staff and divisions within the DEC, Livingston County Department, and Canisius Lake Association, to name a few. We were working hand in hand out there pulling vines, actually doing surveys on adjacent properties and within the same property that was originally found. We treated those sites through mechanical hand pulling several times throughout that first year, and we did a pretty extensive survey of surrounding properties. We used volunteer events once or twice, and then everybody that participated with us was technically volunteering as well, some on the job, some off the job. The big takeaway of that story was just how much cooperation we had that first season, and it was very effective at controlling this initial infestation. In addition to the actual control treatments, we did what I started to describe in the previous slide, which is survey the surrounding properties for the delimitation process. To delimit an infestation like this is just to map the full extent of the invasive or the pest that's new to the area. So that's what we set out to do. And the first step was preparing, using GIS to kind of find out exactly where it was, properties were surrounding, how to contact those landowners, and then acquiring permission. Then we started coming up with some ideas on how we were going to survey, and then figured out how to train volunteers that participated with us. It was all on the fly. Once survey began, we didn't really know exactly where it was going to take, where we'd likely find it, so we looked everywhere and found that it was mostly on the edges of fields, right on that forest to field edge line, or on shrubs on the edge line, or sometimes shrubs in the interior of fields where it was kind of a meadow, transitioning meadow into early successional forest. That became where we found most of the actual plants, and that's how we started to focus. We would do grid searches in relatively open fields where there was brush here and there. We'd do grid searches in openings in the forest where there was a clearing for one reason or another, logging or just blowdowns. And then everywhere else we focused on edges, trail, and then trails as well through the forest. There was enough light hitting those trails. We documented new finds and addressed them as we came across them, pulled plants as we found them. All right, management in 2018. We had a lot of time to think about what we did the first year over the winter, and then we got into the new year in January, and we go, uh-oh, it was good and bad. We found a second infestation quite a distance away in the town of Oakfield in Genesee County. The good thing was that it was found that season. There's a couple of staff from DEC Wildlife that were looking at one of the wildlife management units called Oak Orchard in the town of Oakfield. They happened to notice this funny looking vine that you could kind of see draped over top of the vegetation or the forest edge there. That's John Mahoney, I believe, at the time, and Emily Bonk was taking the photo. Those two foresters realized that it was different than anything they'd saw before. They came to me because they had an inkling that it might be mile-a-minute. That's what they came down to on their own. And we later determined that with the help of PRISM groups and some folks in Albany. Anyway, we now had this much larger problem. This more than doubled the area of infestation. It was far away. We didn't know the source of mile-a-minute, how it got to each location, and we didn't know if they were interrelated. So we had to adapt the plan from the previous year. Thought we were only dealing with Geneseo, not so much. So we hit the ground running as hard as we could. We were able to hire some interns from SUNY ESF. They turned out to be excellent workers for us. Brandon and Lydia, they're pictured in the far right. Their complete focus that summer was on mile-a-minute management and mile-a-minute outreach. The three broad goals were delimiting the MAM populations at both the Oakfield site and Geneseo, applying control techniques, and then getting out there and pounding the pavement, knocking on doors, sending out outreach materials, getting involved in presentations, and they did an excellent job with it. Okay, this is to show where the infestations are here in Western New York. So we focused on that delimitation again, and we were looking at both areas, of course. We had to expand the area for the 2017 work that we did in Geneseo because we found infestation areas a little further out from the original property. So we expanded that area. So thank God we had 2 dedicated staff persons for that. And then we also had to draw up a pretty extensive survey around the Oak Orchard area. That was a little bit scary because there was a lot of land there, and a lot of it was different kinds of wetland. You know, we had some open marsh, some shrubland marsh, some very thick areas, and we had no idea if this plant could establish itself in wetlands like that, in marsh areas like that or not. So we looked everywhere, and fortunately we had some assistance from operations, DEC located. There was a small office up there in Oak Orchard, and they were able to provide some equipment. So my staff there were able to actually get out on some four-wheelers and search large areas quickly. Control treatment was mixed between mechanical pulling at Geneseo, mechanical pulling at Oakfield, and then also herbicide treatments at Oakfield. To do that, we coordinated with certified pesticide applicators, DEC staff members from wildlife, some of our wildlife biologists, and the DEC giant hogweed team. We coordinated with both of them. The hogweed team handled the private property infestation across the street from Oak Orchard we found later in the season, and then our own wildlife staff handled the infestation found on state property on the wildlife management area. Outreach and education was real big. Brandon and Lydia were able to work with PRISM groups to do presentations. We got some newspaper write-ups. Folks with IMAP Invasives New York were excellent. They did a write-up for us in their newsletter. Brandon Analytic created some flyers, and Western New York PRISM and Finger Lakes PRISM both helped develop mailings, and we resent those out that second year, 2018, with the group as well. We got to the end of our collective work at 2018 and took a big sigh of relief. I realized that this infestation and controlling and managing mile a minute was going to be a large task, and I needed more help. I contacted Dr. Amatangelo from SUNY Brockport, and she was awesome. She was immediately interested, and thank God she was willing to get involved, and she really took it over from there. I'm still involved when I can be, and I'd like to continue that. But SUNY Brockport is all over this now, and I'll let Dr. Amatangelo discuss it further.
Great, thank you. So, yeah, so there's been a lot of great effort on controlling the populations, which we're kind of coordinating now and trying to make sure that even with potential changing research abilities and atmospheres, we are able to keep doing that this summer as well. So I'm going to talk a little bit about our current efforts and what we're hoping to do this year. So this is a big collaborative project, and there's been a lot of great volunteer work. A lot of people have come together to make materials for outreach and connect me with people that are interested in Mile a Minute. And this could not be happening without everybody coming together to tackle this plan. The overarching goal, we received funding from the DEC for Mile a Minute eradication, control, and surveying. And our main goals are to continue treatment of the existing populations, evaluate treatment effectiveness in our region, continue surveying for new populations, and then also try to understand maybe a little bit about how our populations and our genetic sources in Western New York might be different from the rest of the invasion to the east and south. So in 2019, we set out to continue what Gary and his team had already started. So we wanted to continue treating as much of the Mile a Minute as we could, while also leaving some control areas up until fruiting so that we could collect those seeds for our greenhouse experiments. And then we also wanted to evaluate the comparative effectiveness of herbicide and physical treatments. So at both sites, we did physical treatments where we hand-pulled Mile a Minute. This was in July. And we hand-pulled all of the Mile a Minute from a subset of our plots and bagged the material. We also had plots that were applied herbicide. This is only at the sites in Oakfield, private land, and Oak Orchard. And then we had mixed plots where we included both. What we were interested in is whether or not there was any additional benefit to removing the dead vegetation after using herbicide. We thought it might make it easier to see resprouts if there were any, and just get a handle on what the Mile a Minute had been doing to the rest of the community in those areas. And then after we applied these treatments, Erica, who's pictured, and Hannah, an undergraduate student, revisited the sites every week to 2 weeks, counted and pulled all resprouts to evaluate the effectiveness of the treatments. And during those physical treatments, we were pulling plants out at the roots, but there's always a small chance that you're going to break off at the base of the roots rather than getting the entire rootstock. And when you're talking about the amount of area that we covered, we wanted to see how effective that treatment was. We knew herbicide was effective, but the hope is that physical treatment can also be effective, particularly as we might run into landowners that are not really enthusiastic about allowing herbicide use on their land. So herbicide was applied by the certified technicians for the DEC, the wildlife workers, and it was a relatively low concentration of rodeo with a surfactant. And this is necessary because the way the leaves interact with liquids, you need some sort of surfactant to get the small concentration of herbicide into the leaves. And in 2019, they used about 32 ounces of rodeo in about 25 gallons of spray to cover most of the region in both Oak Orchard and the private land in Oakfield. And of course, we weren't covering the entire area because we had some mechanical plots and some control plots, but this was only about 4 hours, so it's not as difficult as obviously physical pulling is. And here is some of the infestation on the day of treatment. You can see it at least 12 feet in the air up on the bushes. And then 1 week later, as the mile a minute had been killed. And we had been concerned that when you sprayed it, you couldn't reach the upper leaves. That would be a problem, but it looked like that wasn't a problem. It did die all the way up the vine. So here we've got 4 of our different treatments in comparison. And first of all, in the slide to the left, or the picture to the left, I want you to notice how dense that is. That is all mile a minute. And we actually had a hypothesis that mile a minute would not do very well when it didn't have anything to grow on. And one of my students investigated that, and actually what she found was that mile a minute did just as well without anything to grow on as when it had things to grow on. And actually it grew on itself and formed these mats, these bushes of herbaceous material that were probably almost up to 1 meter high. So if it has nothing to compete with on the ground, it is going to do really well. And the reason that this area was so dense was because all of the woody vegetation had been cleared in the hopes of making mile a minute easier to control. And it is certainly easier to see where the mile a minute is. It's all mile a minute, but it appears that that also released the seed bank. So it created this massive growth the following year after the woody material was removed. So the hope is that that will have actually used up a lot of the seeds, and that this year there will be less density coming up. So on the right, you can see our 4 different treatments. So in the hand-pulled treatments, we were able to leave any native vegetation. We had an herbicide treatment. We had an herbicide plus weeder treatment, and then control areas that we monitored and bagged the seeds to make sure that they did not get released or birds did not disperse them. So our main results so far we've had a couple main takeaways here. So here are the 3 different sites: Oak Orchard, Management Area, Oakfield, private sites, and then Geneseo. And the y-axis is the average number of sprouts per 100 meters squared coming back up during the rest of the season. So larger bars means more resprouting. So M are the treatments that were just physical. So that was just hand pulling. And we see that if you just hand pull, you have about 60 sprouts per 100 meters squared coming back at Oak Orchard and Oakfield, and then less than that, about 2/3 less than that at Geneseo. And we think that's because Geneseo has been treated for a few more years. The initial density was quite a bit lower at Geneseo. There is evidence that treatments have been working as we start to exhaust the seed bank. There did not appear to be any benefit to removing vegetation after using herbicide at either site, and herbicide is clearly the most effective treatment and more treatment effectiveness than physical pulling, easier, but perhaps not suitable in all locations. So we didn't have as much time as we'd hoped to undertake surveying. Our funding did not arrive until February of this year, so we weren't able to hire the additional technicians to help us with surveying. We did what we could, though, and we found 5 new populations around the known infestation at Oak Orchard. Those were all removed. So here you can see the areas in red were new infestations, and in particular, the 2 coral squares in the upper right are large enough infestations that we think that that's going to have to be added into our treatment protocol. We've been hoping to actually have the brush hog out to remove the woody vegetation there, but that's not going to happen this spring, this year. So this year, what are we going to be doing? We have fieldwork planned for 2020. We want to see how the population density has changed in our different treatment areas, continue with treatments and monitoring. We'll probably increase the area over which we use herbicide. We think we have a pretty good handle now on how much better it is than physical pulling. And we're going to really increase survey efforts. We also are currently focusing on a greenhouse experiment. We've got seed sources from 4 populations. We have Geneseo, we have Oak Orchard, we have a Long Island population, and then a population from Pennsylvania. And we're growing them all in a variety of different treatments, of different light levels, different temperature, and different moisture levels. You can see the lower picture here. This is just taken last week. So our Mile a Minute plants are quite vigorous and doing well in the greenhouse. They're not flowering or fruiting yet, but we expect them to do so in the next few weeks. And we're interested to see what this tells us about where our plants have come from and how well they're going to do in different types of conditions. Okay, and that's all I have about Mile a Minute.
All right, well, thank you, Katie and Gary, for joining in and discussing all of the Mile a Minute work. Sounds like, you know, there are a lot of ambitious plans for this year and a lot has been done already. And so everyone just wants to get their questions together. You can submit them to the chat and we will take the questions now.
So Steve, I see you just asked about the Mile a Minute weevils, whether we would experiment with them. We've been in talks with people in other locations, and from what we've learned or been told, our populations just aren't big enough to make that a worthwhile effort. I think that if the oak orchard population ended up intractable and we couldn't get it under control, and we continued finding more infestations nearby, then maybe we would we'd kind of go that route. I don't know if Gary has any other thoughts about that. I know that you had talked about thinking about that a bit.
Thanks, Katie. Yeah, we discussed like every possibility, but we came to the same conclusion that you did based on people we spoke to in the Catskills and other people that we conferred with on the matter, and said the same thing. If you're going for eradication, you have to go for eradication. You can't try to run both things at once. You know, the biocontrol is for a population that's established and extensive, where it can exist and can continue to thrive. So we did not want to let that happen, so we let it go like that.
So I see another thought about using flame control for eradication. I have not read or thought much about using fire for eradication of Mile a Minute. I'm not sure if you meant Mile a Minute or if you meant one of the other species that were covered. If Brittany or Gary, you want to speak to that?
I don't have really anything very good to add to that, Katie. Like I know that we did look into it a little bit. It certainly didn't make sense given the location in Geneseo, at least. One of the things that, you know, we learned from this experience is that a lot of what you're going to do for management is dictated by the property owners, and the designation and use of the land. And the concept of using fire or chemicals was not really something that the residents there around Geneseo were very interested in. And we discussed it a little bit more with the folks at our own property in Oak Orchard. And I honestly, I don't remember where we left off on that. It's an open discussion.
And then going back to Slender Falls Brome, I've seen mixed results. It seems like if you use a blowtorch or some sort of small scale method, it's not really effective and it actually comes back a lot thicker. But I know out in the West, they had done some prescribed burns and maybe saw a little bit more effectiveness. And so again, I just don't think it's an option, at least for the populations that we have found and are working with at Letchworth State Park and Genesee County Park and Forest.
So Steve asked, is it possible with Mile a Minute to just cut or mow with brush mower and leave the stems to die? So that's a good question. We did see evidence that if you break off the stems near the base, they can resprout. So we don't know if that's really going to be a great treatment option. And we do have a plan this summer to do a little experiment with that, basically let a patch grow to a robust height and then mow it and then see how much it does spring back. So that's something we're going to try to check out this year.
So it doesn't look like there are any other questions in the chat. One thing that Steve had wanted to point out earlier was just that the Mile a Minute fruits, even when they are still green, still have viable seed. You shouldn't wait until the berries are blue, because at that point they've probably been viable for quite some time. So if there aren't any other questions, yeah, so I guess I'll just say thank you to our presenters, to Gary, Katie, and Brittany. Those were great. I appreciate it.